Imagine a city where streetlights suddenly turn on more frequently. This is what happens when chronic inflammation triggers microglial activation in the brain. Below this threshold, microglia remain dormant, like a pressure valve sealed shut. Above it, they lock open and release an uncontrollable surge. Chronic inflammation elevates Compensatory levels, pushing them past the regulatory boundary. Microglial cells, normally dormant, become hyperactive. They overprune, disrupting neural networks and leading to cognitive decline. The brain's intricate balance relies on these coupled thresholds aligning within a tight tolerance. When inflammation tips the scale, microglia overreach, and healthy synapses are mistakenly tagged for removal. This mechanism, once protective, becomes a source of neural disruption. What survives this pruning is not random. It's determined by the alignment of these thresholds. But what decides when they fail? The answer lies in the intricate dance between inflammation and microglial activation. 2018 used PET imaging to peer into the brain's hidden dynamics. They found a significant increase in microglial activation in inflamed regions. Imagine streetlights turning on more frequently, signaling increased microglial activity. Below this threshold, microglia remain dormant, like a pressure valve sealed shut. Above it, they lock open and release an uncontrollable surge. As we delve deeper, we uncover how this heightened state affects the brain's delicate balance. Jones et al. Used in vitro models to study microglial behavior. Below a certain inflammation threshold, microglia act like streetlights off at night, dormant and inactive. Above it, they become hyperactive, pruning synapses at a rate significantly higher than in control conditions. Imagine a garden where pruning shears are overused. Below the threshold, the shears remain locked away, but above it, they are constantly in use, cutting more than necessary. This excessive pruning by activated microglia disrupts the brain's delicate balance, leading to a loss of important neural connections. This overpruning sets the stage for further neurological consequences. 2019 used advanced MRI to study Alzheimer's patients. They found a significant reduction in neural network connectivity in affected regions. Imagine a city where half the roads are closed, isolating neighborhoods and disrupting traffic flow. This is what happens when microglia overprune synapses, severing vital connections. In the brain, these synaptic highways are essential for information transfer. When microglial activity exceeds normal levels, it acts like a pruning tool that cuts too deeply, leaving the network sparse and inefficient. This excessive pruning disrupts the intricate web of connections, leading to cognitive decline. The next threshold will reveal how the brain attempts to compensate for this loss. 2017 used protein expression analysis to uncover a critical shift in brain chemistry. Compensatory levels increase significantly in inflamed brain tissue. Imagine sticky note tags that mark items for removal. These tags are the C1q proteins. Below this threshold, healthy synapses remain intact and functional. Above it, these tags label even healthy connections for destruction, triggering microglia to prune synapses excessively. The next threshold will reveal how the brain's compensatory mechanisms fail to restore balance. 2021 used a microglial activity simulation to reveal a critical boundary. Below the regulatory threshold, microglia function like diligent gardeners, pruning only damaged synapses. Above it, their activity rate exceeds normal levels, leading to excessive pruning of healthy synapses. This overactivity means microglia begin to prune healthy synapses as well. Below the threshold, synaptic balance is maintained. Above it, the brain's compensatory mechanisms fail to restore this equilibrium. The boundary marks where the brain's natural checks and balances break down, leading to excessive pruning. This threshold reveals the precise point at which chronic inflammation triggers microglial overreach. In this state, the brain loses its ability to distinguish between healthy and damaged connections. The result is a cascade of synapse loss that disrupts neural networks. This boundary sets the stage for understanding how chronic inflammation can lead to cognitive decline. This heightened state of microglial activity not only disrupts neural networks, it also leads to a cascade of further neurological consequences, such as cognitive decline and memory loss. The brain's compensatory mechanisms are overwhelmed, leading to systemic failure in maintaining synaptic balance. The stakes are high. The brain's ability to function is at risk. Chronic inflammation disrupts more than just neural networks. It triggers a systemic response that can lead to widespread cognitive decline and memory loss. Microglial overreach, once initiated, becomes a self-perpetuating cycle, further damaging the brain's ability to heal itself. This biological cascade highlights the critical need for maintaining the delicate balance of microglial activity. The precise alignment of these 5 thresholds, chronic inflammation, microglial activation, excessive pruning, connect tagging, and regulatory failure, reveals the narrow tolerance required for maintaining neural health. Each threshold acts as a checkpoint in the brain's intricate defense system. Below this boundary, synaptic balance is maintained through a delicate dance of growth and pruning. Above it, the checks and balances break down, leading to excessive synapse loss that disrupts neural networks. Chronic inflammation serves as the initial trigger, setting off a chain reaction. Microglia, the brain's immune cells, become overactive in response. They begin to prune not just damaged synapses, but healthy ones as well. This overactivity is exacerbated by connect tagging, which marks synapses for removal. The regulatory mechanisms that normally keep microglial activity in check fail, leading to a surge of excessive pruning. The brain's natural resilience depends on these thresholds working together. When chronic inflammation breaches the first threshold, it sets off a cascade of events that can overwhelm the system. Microglial overreach is not an isolated event, but a consequence of multiple failures. Each step in this process highlights the critical balance required to maintain neural health. The joint improbability of these thresholds aligning perfectly underscores the brain's complexity and fragility. This narrow margin for error means that even small disruptions can have significant consequences. Understanding this mechanism is crucial for recognizing the delicate balance that must be maintained to prevent cognitive decline. By visualizing the brain's defense system as a series of interconnected checkpoints, we gain insight into how chronic inflammation can lead to microglial overreach. Each threshold represents a critical boundary where the brain's natural defenses either succeed or fail. This mechanistic understanding reveals the elegance and precision required for neural health. Other thresholds may exist that interact with chronic inflammation and microglial overreach, further complicating neural health. In our next episode, we explore the role of neurotrophic factors in maintaining synaptic balance. Stay tuned to uncover how these growth-promoting molecules can counteract the effects of excessive pruning.